Okay. Thank you all for being here. Welcome, welcome back. And I just want to thank our speakers already for this morning. We've had an amazing morning, haven't we? Yeah, it's been amazing. Yeah. Thanks, everybody. And we have an amazing lineup for this afternoon. So I want to start with introducing our keynote speaker for the afternoon. And I think we've already heard his name maybe once or twice this morning. Coming all the way from MUSC, from the Medical University of South Carolina, we have Dr. Russell Chip Norris here today. So let me just tell you a little bit about Dr. Norris. Dr. Norris is a professor of regenerative medicine and cell biology at MUSC. He has clinical appointments in both neurosurgery and in cardiology. His research basically focuses on genetics and connective tissue diseases, with publications in leading journals such as Science and Nature Genetics. He is internationally recognized for his work in mentorship, and he is paramount in moving the research field forward in patients with Ehlers-Danlos syndrome and other hypermobility disorders. And I just want a little welcome for Dr. Norris, so please welcome him.
All right. Well, what a pleasure to be here. It's tissues. I get a little emotional sometimes, so I apologize in advance. It's really a pleasure to be here. This is just an amazing institution. I've known Lori, Dr. McMahon, for many years as our former VPR. And just what they're building here is incredible, and I look forward to working with them for many years to come. So I don't really like my title. In fact, I really don't like titles of talks at all because they always seem corny. So I'm going to move on. This is what really matters. This is my group. It changes a lot because we have a lot of different people come and go. I don't know how many, there's maybe 30 people in here. 16 of them have Ehlers-Danlos. We hypermobile type. We are committed to including these patients into our programs. And as you heard Molly just talk about, it's really an amazing program. And I'll talk about it more a little bit later. I have one slide on what EDS is, or maybe two slides. It's characterized as and this is like, who doesn't know this? It's characterized as a genetic connective tissue disorder. I would argue that it's not a primary connective tissue disorder, but that's up for debate. Significant healthcare burden. Everybody in this room knows that. Very little is known about the phenotype and the genetics and the biology. And there's obviously challenges because of this in diagnosis and treatment. Phenotypic heterogeneity is the rule. And defining HEDS is really restricted to a clinical diagnosis. These are things that are known. So let's try to figure out things that aren't. What's the problem with physicians? Yeah, sorry, sorry. There's even at MUSC where we have a really we have a lot of physicians that actually understand and recognize this disorder. But unfortunately, this is how it's taught. This is a slide that comes from our own medical school teaching curriculum in the musculoskeletal block. This was 3 years ago. So if you're a medical student learning about Ehlers-Danlos, learning about musculoskeletal conditions, and this is a slide that you're presented with, how do you take this? You take this as, oh, it's rare, and it's, they have stretchy skin, they might bruise, and, they certainly don't have a whole lot of other things wrong with them. So if you see these patients and they have a litany of issues, it must all be in their head. And that's what they're taught. And this is a real problem. So this is no wonder why physicians don't care. Physicians in this room do care. That's why they're here. But we need to educate the physicians as a primary care physician in Fargo, North Dakota, that when a patient comes into his clinic, he understands and knows what this condition is. And we have a job, and albeit a responsibility, to pioneer this. And I think with UVA, I think we have a real chance to accelerate this. and I'm going to talk about some ways that we can. This is what HEDS really is. This really stemmed from Dacre's work, as well as a couple of recent papers from us. You'll see I'm not going to go through this. There's a lot of comorbidities and conditions that are wrapped into what HEDS really is. So there's a great disconnect between clinical diagnostic criteria and reality. I think everybody recognizes this now. And we really do need to do better for these patients and their families. And so this is sort of how I view my lab. Basic science research. I'm just a dumb scientist. I'll talk about like why me. I really don't want it to be about me, but I'll talk about how I got started a little bit later. So I'm a basic scientist, so I'm really interested. I got trained in molecular genetics, and I'm really interested in genetics and biomarkers and developing models and mechanisms, models that will lead to mechanisms, and then that leads to therapies. Very interested in clinical research, education, and then of course advocacy and awareness. This is what my lab does. I just sit behind a desk and try to think of things to stay creative and passionate. So how do we start? HEDS is a really tough thing to start with because it's, where do we go? What's the first step? And so the first step for us was this: IRBs. I hate IRBs. I'm a scientist. A scientist writing an IRB is just a terrible idea. In fact, my IRB, finally, after I'd done like 35 revisions of my first genetic IRB, said, why don't just hire a coordinator to write this, because you're terrible at it. So I did. I actually got Dan Judge, who's a cardiologist, helped me with it, and then we did it. We got the genetic registry approved. Now we have 18 different IRBs. We span almost every type of, or a number of different specialties, including orthopedics, neurosurgery, GI, autonomic dysfunctions, musculoskeletal, genetics, biomarker studies. So, I mean, we needed these. Now we didn't need all these. We just needed an umbrella one, which is what we're doing now. Is just having a big umbrella IRB that's going to take place of all of these other ones. But this was really the starting point. Okay, we need to start with the patients. We need to start collecting clinical samples and see if we can understand anything from the patient, because there weren't any models. There weren't anything to really go for. So what does this allow us to do? It allows us to, collect samples. And now we have, I'm also the director of, the new scientific director of Precision Health at MUSC. I'm really happy about that job. But it does allow us to develop these biobanks. And we have an amazing biobank. Now we have over about 7,000 biospecimens from EDS, and it actually is supported through the entire institution. So costs are offset by the medical university, which is an amazing way to do it. So that really allows us to do a lot more than we previously could. So where do we go from here? We first wanted to better define HEDS. Really put numbers to it, really put metrics to it. And that started with the survey. Not dislike the one that Dacre and his people did at Mayo. It's very similar, and very similar results as well, which is great. And so we had, in the end, after we filtered down through all these different criteria, 3,300 in the HEDS group and 546 in the HSD cohort. And this is a problem. This is something that's really continually mentioned, and is a limitation of some of our studies, and really everybody else's studies, is the high percentage of Caucasian females in all of our research. I mean, it's 90. In ours is 95.8% white, and so the people of color We need to figure out how to do better with them, and hopefully we can develop programs that will actually highlight their individual needs. In our study, we identified this diagnostic timeline. This was known, but we knew it was like long, but it was really profound in our survey. I mean, 22 years. I mean, that's just, how do you live with that? You have to do better. And then the comorbidities, profound, right? You guys know this, and you saw this panel in B earlier from Molly's, but I think what really was interesting to us, and it was something that we kept hearing from the patients, is that it wasn't necessarily so much the joints and you hear collagens everywhere. Yeah, of course it is, but what we were hearing, the major things that were wrong, or that these patients were really were chronic pain, autonomic dysfunction, and GI. Okay, joints was fourth, and that's telling, because when I started this, I was always thinking that this is Ehlers-Danlos. This is a collagenopathy problem, primary defect in collagen. Who came up with that decision? Just because people are bendy, you assume that they have a primary collagen disorder? Where's the data to tell me that? So it started me thinking, like, questioning that as a primary cause, and maybe it is, but maybe it isn't. And I think we need to leave that option open. This was really confusing in the context of a primary collagen condition, although patients with Marfan syndrome and Loeys-Dietz and OI are known to have some triggering events, but 70% of our survey reported a triggering event. And these range from puberty, viral bacteria infection, physical accident. These are all things that engage the immune system. And that's what really started trying to, speaking to me that this may be more complicated than a initial problem with collagen. So the population demographics has really helped provide a better definition to HEDS, and I think that this should be included in any sort of diagnostic criteria that's being developed. Is it really a subtype of Ehlers-Danlos syndrome? That's not for me to decide. Other smarter people will decide that. Trigger events that I just talked about. The pain, GI, and autonomic function, and the immune system really made us think about mast cells. I mean, you've heard that now, which is crazy, a few times already this morning. And maybe molecular analyses will help. And so I'm going to tell you about this candidate gene that we identified, and maybe hopefully try to clear up some confusion. We had a large family with inherited autosomal dominant HEDS. These are clinically defined by 2017 criteria. We did whole exome sequencing on the two most distant relatives, and that's presented here with the arrows and the arrowhead. And from that, they shared 48,000 variants. We then filter that based on standardized filtering criteria, and identified 6 rare and potentially damaging variants. Only one of these variants tracked with the family. So this is the only thing in the coding region in this entire family that tracks. It's pretty good. This is the variant we found is in KLK15. Many of you in the room have probably read this paper. We identified a second family. It's just a trio, and then the population frequency. And here's where some questions come in. CAD score tells you about the severity of the mutation. That's pretty high. The range goes from 0 to 35. 24 is known to be probably pretty deleterious. The minor allele frequency, though, is in NOMAD is 0.002. This means 0.2% of the population have this variant. Seems too common. And it is too common to, in my opinion, to by itself cause the condition. But maybe not. But is this a standalone genetic paper? No. in order to confirm pathogenicity, one thing we always do is actually generate animal models. And looking at the mutation itself is a disservice to the study. And we need to recognize the body of work for what it is. And so we made a mouse with the mutation. Now the mouse that has this mutation, that's the only thing changed in this entire animal. So any sort of phenotype that we see is directly caused by that change. So we did mechanical testing. I'm not going to go through this. There's statistically significant values. What this all means is that the mutant mice have increased elasticity in the toe region of the stress strain curves. I'm sure it's like you all know what that means. This is actually this is on an Achilles tendon that was stretched. Okay, and the curves represent the stress strain curve. And in the end, what it means is that the tendon and ligaments are more elastic. Okay, so it's a functional defect. That's good. That's caused by that variant that we put in. Structurally, we looked by transmission electron micrographs. And what you see is collagen fibrils. Those are those little round circles. That's what your collagen fibrils actually look like in your tendon. This is what it looks like in that mouse. Same region of the tendon, same analysis. You'll see that there's a lot of little circles in there. They're not supposed to be there. We can quantify that. We've quantified almost 8,000 of those. It's painstakingly was done by 3 independent reviewers, all blinded to genotype. And you see that there's a leftward shift, meaning on average, the fibrils are smaller, and that corresponds to a more elastic tissue. So there's a structural phenotype in these mice. What about functional? And so we did echocardiography. We knew in the family that there was mitral valve prolapse as a common cardiac phenotype. This is a control mouse. Everybody here is, I'm sure, an expert on ultrasound echoes. The mitral valve is located right here. Yeah, everybody can see that clearly. Here it is in a blown up version. And in the KLK15 mutant, you'll see very thick tissues here, and they're actually prolapsing into the left atrium, showing that these mice have mitral valve prolapse. This is shown on higher magnification here. 83% of these mice have valve dysfunction, whereas 0% of the controls did. Okay. Histopathology, you see that they have myxomatous valves. So this really validates the mutation pathogenicity, even in the case of it not being enriched or having a more higher minor low frequency than one would expect. We then did whole exome sequencing on 197 clinically diagnosed HEDS patients. This was, here's a list of, and we wanted to scan KLK genes. By the way, kallikreins are what's in snake venom that actually makes you anaphylactic. So there's something interesting with what Anne was talking about before. You don't want to screw these genes up. It's also involved in blood clotting, which is not surprising, I guess. This is a list of all the KLK mutations. The KLK is a gene cluster of 15 genes. They're all organized together on one chromosome. And this is all the mutations that we find in these patients. There's a lot there. We did TRAP-D analysis, which is just a fancy term for saying, when you take these individual genes and the variants we find in each individual KLK gene, are they enriched in the population? And the answer is, you'll see the asterisks. This is per gene, so not per specific variant. You'll see that there's statistical enrichment of KLK, specific KLK gene mutations or variants in HEDS compared to controls. Be nice to have this. Should be pretty easy to validate this. So there is statistical enrichment. However, the KLK15 variant, the one that we actually made the mouse of, is not enriched, and we know that now. So that's a little bit confusing. But when we look in the literature, and this is primary literature that's not older than 5 years other conditions that have minor allele frequencies higher than expected and are not enriched in the population, but are known to be pathogenic. And this is just a list of probably close to 100 different conditions. So you see the disease over here. You'll see these are all missense changes for the most part. There is sex bias on most of them, and they all involve triggering events. And they all have high CAD scores and relatively benign minor allele frequencies. So, in here are a lot of different autoimmune conditions. And so, I think that that's something that needs to be taken into consideration and not just a blanket not enriched in the population. In some diseases, like autoimmune conditions, this is actually not unusual. It's more the rule. And the reason is because of incomplete penetrance, strong dependence on genetic background, environmental or immune trigger dependence, context-specific molecular effects, protective modifiers, and population stratification or ancestry effects. These are all things that are already well established. And it's interesting that HEDS, what we're finding out about HEDS, it actually fits in these boxes pretty well. So a pathogenic variant may be pathogenic without being enriched in broad case controls cohorts. Regardless, this does not mean that the KLKs are the only cause of HEDS. In fact, they're going to be a minor contributor, and it's likely polygenic. And so how can we test this? We do a genome-wide association study. And it's basically taking a group of EDS patients and a group of controls, doing a genotype study using SNP arrays and seeing if there are certain SNPs that are more prevalent in one population versus the other. So we did that. This is the design. We did a 3-case control study. There are 2 cases, 860 cases in case control 1 from MUSC, 493 from case 2, and then 462 cases from all of us case in the GWAS case 3. These are case 1 and case 2 are self-report HEDS. And case 3 is EHR confirmed physician entered 2017 criteria. Well, they don't say 2017 criteria, but physician entered HEDS diagnoses. We then go through and identify through a very complex set of experiments that takes a long time to do. We identify whether or not there's actual signals. One of the first things that we did was when we got all the genetic architecture back of these 1,800 EDS patients is to do a genetic correlation between other known conditions where we could have summary statistics. And so we could compare, like, how similar are our genetic findings in HEDS with those that have already been done and published and publicly available. And so what you can see is sort of unsurprising, but nice to validate our study is ME/CFS has a similar genetic architecture, chronic pain, fibromyalgia, IBS, GI issues, migraine, etc. So this was really nice that, support that we were on the right direction. And then the Manhattan plots. These actually tell us the specific SNPs that we see enriched in our population. What you'll notice is that if you look at the GWAS, the first rainbow colored thing, there's only 2 things. There's only 2 SNPs. And this is just a reflection of the disease heterogeneity. Had we not done 1,800 patients, we probably wouldn't have found anything. But we did find these 2. And then we did what's called a GBAT, or gene-based association test. And instead of using individual SNPs, we said, well, let's use all the SNPs that we identified in the entire gene and see if that's enriched compared to controls. And then that led us to an additional 5 genes that are likely involved. And then we did a TWAS. I know these are really complicated terms. And anyways, we found a few more genetic signals that are likely relevant to the disease presentation. So I'm just going to look at ACKR3. That's a weird slide. That's sort of our lead hit. This up here is our locus zoom, We have a lot of different SNPs in here that builds the support for this being a true signal and not a false positive. EQTL, meaning the variant is actually expressed, and we see it expressed in tibial nerves, and then also in the by doing ATAC-seq and single cell ATAC-seq and looking at the status of the chromatin around that SNP, we see it's an open chromatin region, meaning that it can actually be involved in transcription regulation. The SNP actually changes a binding site for an important transcription factor, which was interesting to me. It goes from not being a transcription factor binding site to one in which a transcription factor called aryl hydrocarbon receptor binds. And we validated that this actually happens through reporter assays. What's interesting about this is that AHR, or aryl hydrocarbon receptor, is stimulated by environmental stimuli. So it can be environmental, it can be toxins, pollutants, viral infections. Its activity is increased. And maybe patients with HEDS are more susceptible to responding in a different way to these sort of pathogens, like Dr. Maitland was saying earlier. So what is ACKR3? ACKR3 is a receptor, which is nice because it can be targeted. It is involved in opioid clearance. It's a pain receptor in the brain, which to me is very interesting that maybe we've identified some reason for why patients with HEDS have more pain. It actually sequesters and degrades endogenous occurring opioids in your brain, disallowing the opioids to act on endogenous opioid receptors and giving you analgesia. So we see increased receptor and therefore likely increased clearance of opioids. It does a number of other things. So it does pain and is involved in neuroinflammation. It is highly expressed. In fact, probably one of the highest expressed tissues or cell types are mast cells, which is interesting. We're really looking at doing that. It is looking at furthering those studies. It's involved in connective tissues, GI dysfunction, barrier function, as Dr. Maitland was saying earlier, epithelial repair. Like this thing sits at the center of a lot of things that make sense. Blood pressure regulation, anxiety, depression. This is what it does. It regulates these things. In fact, if you just infect a mouse with ACKR3 receptor, the mouse becomes incredibly anxious. So just that alone is really interesting. So we're now developing in vitro and in vivo models to look at this pathway. It's a pretty cool pathway. But what about changes in the blood? And so we did an unbiased mass spec approach. And we, on 29 different individuals with confirmed 2017 clinically diagnosed HEDS and age match, sex match controls. And what was interesting about this, and this was recently published, was that really out of the 45 genes or 45 proteins that were differentially expressed in EDS patients versus HEDS patients versus control, they really fell into two main categories. Kallikreins, which sort of seemed to make a little bit of sense with our genetic work, as well as the complement cascade, which Dr. Maitland talked about earlier. And then we did, this is just a heat map over here on I think all of the patients, showing that there is a loss of complement protein. This is known as complement consumption. It's activation of the complement program. I'm not going to go through what it is and talked about the complement system a bit. We then validated this using specific ELISAs for individual complement proteins. So now we have really essentially a 2 or 3 tier approach validating our studies on complement, and complement sits at the top, or is one of the top regulators of the immune system, upstream of mast cells. So I think that we did not find any changes. We looked at collagen and fibronectin because it's a connective tissue disease. We thought, well, maybe we'll find some things with collagen and fibronectin. We see fragments of collagen and fibronectin, but there is no difference. We detect no difference either by mass spec, which I'm not showing here, or by Western analysis. And we did the Western analysis 3 different ways to confirm that what we were finding was correct. So for an overview of this summary, we established a large clinical and genetic registries with the goal of better defining HEDS. Some of these studies have been published, so you can, if you're interested, read them. We identified multiple families with that single variant that tracked with disease, and then the in vivo data validated the pathogenicity of that mutation. And then we also did in vitro data that I didn't show you here that's in the paper to show sort of some of the molecular mechanism of how it works. Clearly, it's not responsible for all of HEDS, and it should not be used currently as a molecular diagnostic. I think this is really important. Don't ask your primary care or your geneticist to use KLKs as a screening tool for defining HEDS. It's not meant to do that right now. Biomarker analysis identified the immune system and complement. This really, all the data that we have collected so far, really starting from the patients and for them telling us about their condition, questions whether or not HEDS is a primary connective tissue disease, or is it rather a disease that's rooted in mast cell neuroimmune dysfunction? And if I had to bet, I'd go that way. Maybe it's both. Maybe it is. One thing that's but we're betting that mast cells are really important. And everybody, not everybody, but most people think of mast cells as these little sacks of granules that spit out tryptase and histamines when they get angry, and yeah, it's true, right? They degranulate, and that's a problem, and people try to get on mast cell stabilizers. And so we did the experiment where it's a simple experiment. We took fibroblasts and put them in a dish. They stick to the dish. Then we threw mast cells in. See what happens. In 30 minutes or less, Roman was telling me in like 0 minutes, the mast cells are pulled to the fibroblasts. There's a physical contact made between fibroblasts and mast cells, which is absolutely stunning. And you can see that here. So we have ways of labeling these cells. Green cells are fibroblasts. Non-green cells are mast cells. And you can see the mast cell right here is sitting on top of the fibroblasts. This is an active process. The mast cell needs this, the fibroblast needs this mast cell to come close, and it does so rapidly, which tells me it's doing it for a reason. I don't know what the reason is, but I think it's interesting to look at. And you can see here, we've labeled the mast cell red. It actually goes around the fibroblast, and it has these little spikes. I don't know if you can see these on the screen, but these little spikes that attach, sort of anchor into the fibroblast. So we're very interested in looking at mast cell and any other cell type interaction because it hasn't been really looked at. Mast cell fibroblasts, of course, putting them together and then asking how does their interaction change each other when they're not there. What's their transcriptome? So we can do this. We can actually look at every single cell, every single mast cell and every single fibroblast when they're together and say, when they're together, their entire transcriptional program landscape has changed. What has changed about it? And versus, hey, if we just degranulate some mast cells and throw that degranulation stuff on a fibroblast, how does that change the fibroblast? I bet we're going to find that the connection between these two cells is just as important as if they degranulate and release histamines and tryptase and prostaglandins, whatever else. But that's not known. But we're going to figure that out. And we're going to figure that out when we look at mast cells and macrophages, mast cells and neurons. Hey, that's going to be really cool. Mast cells and endothelial cells, looking at blood vessels. These are tissues where the mast cells are there. So what are they doing? This is in the absence of degranulation. This is a 3D image of this projection. I call it a fiber tentacle. I make up words when to try to. We've gone back and forth about a lot of different terms, but it does. These fibroblasts shoot out these almost looks like, you know, an octopus shooting out this tentacle with a suction cup, bringing it back, and you can actually see this in real time. This right here is a mast cell has already been brought into the fibroblasts. And it's being encased in different types of proteins. And now it's going after this mast cell. This happens in 30 minutes. This is incredibly quick. So the single cell RNA-seq will help. The functional aspect of this is actually the consequence of this. I don't have time to go through this assay, but the functional consequences, it actually changes. This interaction changes how the extracellular matrix looks. That's really important. How can we do more for these patients, though? So that's the sort of the sciencey aspect of what I'm going to tell you. We need to build EDS research communities. We have 5 pillars: lay public presentations. You know, I have people in my group that do this really well. Engaging Congress and NIH, building larger research communities, modified medical school curriculum, and an intern program, which Molly talked about. I'll talk about the bottom 4 of these. First, patients are, you know this, patients are the true experts and have unique perspectives and research insights. So engaging Congress. This is actually, Lori was a champion of this. And this is really needed. You need those leadership champions that will go to the leadership at your hospital system and lobby for wording to get into programs that then universities level lobbyists can go to the government and pitch. That's the way. That's a great way to make change, and that's how the voice gets listened to. And this was actually from 2025, I guess. Some of this wording that we proposed. This was, I guess, maybe after Lori had left. She was the pioneer. She made this happen, and then we've since made this happen every year since. And it's a call to advocate. It's a call for NIH, for Congress to approve funding to research groups around the country to have money to study this condition. And this is, you can read the wording. I'm also happy to send slides to whoever, and you can read this. It's, you know, so important to have this. And since then, I will say that National Institutes of Arthritis and Musculoskeletal Systems Institute has actually made Ehlers-Danlos a research priority. So this matters. And you can do this here. I hope she will, just to make her. And this needs to happen across the country. So we can't solve this problem alone, clearly. We need to build larger research communities. Our goal is to increase research engagement from scientists and physicians. I don't want to be the only group to do this. I need other people. There are 10,000 scientists and physicians studying connective tissues, but nearly none of them work on EDS. This is a problem, right? I mean, come on, 10,000? We can't solve this problem? Engagement, tools, and money. That's the issue. What if we can remove one of those issues? And so we started a bold vision where we wanted to develop an EDS toolbox. And this is for all the other subtypes, the rarer subtypes. We propose to generate CRISPR knock-in models for every subtype of Ehlers-Danlos syndrome and offer them free to the global research community. We would do this for free with the goal of building those communities in institutions across the country and across the world, so that those 10,000 physicians and scientists have a tool to study. And we've completed this, which is incredible. This was recently completed. We've now started sending these model systems out across the country. I will say that one of the first ones we sent out was the cardiac valvular EDS, which is exceedingly rare. The person at Case Western received the mice, worked on the mice for a little bit, and then wrote a grant and got it funded for $400,000. Now his lab is switching from a solely, he does valve work, so that's why he wanted the cardiac valve disease model, to including other types of rare EDS subtypes. $400,000. That would not have happened without this. And we had over 500 inquiries or web checks to be engaged with other scientists from around the world. That's really cool. Can we do more? This is that slide. Obviously, that's a problem. So last year, after a lot of work, we succeeded in changing the medical school curriculum in South Carolina. That's not an easy thing to do because medical schools teach to the boards. Ehlers-Danlos is not on the boards. Marfan syndrome is, but Ehlers-Danlos is not. Medical schools will not change. It's really hard to get them to change their curriculum if it's not on the boards. But if you push AAMC and develop strategic partnerships, they're the ones who actually make the board questions. Get them on board, and you change curriculum across the United States. That's where we need work, because that's where you're training your next level of physicians. Start there. Am I going to change the opinion, regardless of what your diagnostic criteria is? Am I going to change the opinion of a 60-year-old physician working in rheumatology that only wants to see scleroderma? No, I'm not. Not going to do it. But if I get these physicians aware early, they're going to see these patients and they're going to start thinking about it more. So this was feeding on that, like, how do we change the perception of this disease? And Molly really spent, a lot of nice time talking about it. This to me is my most important legacy, I will say. That it was really a brilliant idea, maybe my only good idea I've had. It really was a great idea. And it started with a $50,000 donation that we then turned into a whole program that's trained 150 patient scientists. It's incredible. So that's really, that will be my biggest legacy, regardless of whatever we discover. So let me go here, and then I'll finish with some final thoughts. Nothing of significance happens alone. This journey for me with Ehlers-Danlos started 7 years ago when Courtney walked into my office and changed how I view research. And so there's a lot of people here that need to be thanked. A lot of funding societies. What's not on here is the Connective Tissue Coalition, which has been very, very helpful in our work. Of course, the patients and the passionate donors. Without this, none of this could happen. So I just want to leave you with a few final thoughts. It's really an honor to be here. I don't like the publicity. I don't like people knowing who I am. I don't like it. But it's okay. I don't normally script anything, and my lab gets really worried when I go off script. So so if you see me looking down, I'm reading from my notes, because I think it's important to get this right. It tells you a little bit about me. when I was in high school, I was a good student. I studied hard, but I was a really bad student in biology. I almost failed biology in high school. And in some ways, I was a bit lost. It was a big challenge to me. Biology, I just didn't get it. And yet somehow I went on to major in biology in college. And my parents thought I was insane. Like I was going to be an engineer, and I didn't want to do that after, because I thought it was a challenge. I didn't want to be bored in my career. I wanted to be challenged. And then I pursued a PhD, which was even more insane, and then stayed in academic science, which academic science is really hard to get into and really hard to stay in. But over the last 31 years that I've been in academic science, this work that I do has really challenged me, shaped me, and humbled me in a lot of ways. One of the greatest privileges of my career began about 7 years ago, obviously sparked by Courtney, who many of you probably know or follow. And she opened the door to a different way of thinking. One that wasn't rooted in just hypotheses or experiments, but in listening. In listening to patients, in listening to their stories, and allowing those encounters to shape the questions we ask. That to me is one of the most important lessons of my scientific life. And it's something that most scientists fail to even acknowledge. Almost every scientist I've talked to has never met a patient, yet they work on clinically relevant conditions. Patient stories do more than just inform us, they connect us. They create moments of real human understanding. They remind us of what matters. And if we're paying attention, they teach us how to ask better, more meaningful questions. So while we may be all in a different place in our lives, in our careers, in our health journeys, we share something essential in this room. We share the opportunity to work in a world of ideas where empathy fuels imagination, imagination fuels passion, and passion fuels discovery and hope. Sometimes we make discoveries, and sometimes we make a difference for patients. And if we're really fortunate, sometimes we do that at the same time. But success in this work also comes with responsibility to support each other, to lift others up, to create a space rather than to close it off, to build connections rather than fracture them. I don't care who makes the first discovery for something. What I care about is how that discovery can be applied to the patient who's suffering. I care that there are still far too few people studying these conditions. And when too few voices are engaged, the narrative gets controlled by the loudest few. I don't want to be that. That's why I started the whole thing with the mice, to try to get more science out there. This is not how progress happens. That is not how science should serve people, and that is one of the reasons I care deeply about bringing scientists, more physicians, and more thinkers into this field. Now, I went to an all-male, so during high school, it was an all-male Catholic high school. Sorry, I'm telling you this because people always ask me about my backstory. So I went to an all-male Catholic high school where the motto was "Men for Others." And really, that idea has stayed with me my entire life. It has become a kind of compass for how I live, or how I want to live, how I want to lead, and what I hope my legacy will be. I want to keep the door open for the next lost soul who's failing biology in high school, or is seeking a place to belong for a purpose. That belief sits at the root of who I am. I'm incredibly lucky to do the work that still challenges me, still inspires me, and still feels deeply worth doing. I'm not going away, I don't think, unless I have a major health event, and then, yeah. I don't take it for granted, and I'm continually humbled by the trust that this complex, misunderstood, and too often overlooked patient community has placed in me and my group. I want them to know that our commitments in my group and the groups that I hopefully will form have never wavered. Our mission is not driven by money, by popularity. Certainly not. I hate being popular, or by controlling a narrative. It is driven by the desire, the true desire, to help, and by the conviction that patients deserve better answers, better science, and better hope. So thank you, Dr. Knight and Dr. McMahon, for inviting me to speak about this work, which is at its heart the patient's work. Everything you saw was from patients. So cool. Your vision to build an institute here at UVA stands as a real beacon of hope, and I'm genuinely excited about that opportunity to learn from you, work alongside you, and help build something meaningful together now and into the future. Thank you.